In this video, we're going to talk about genetic differentiation, but mostly mutation and why mutations are important to study. So the first thing that we're going to talk about is cell differentiation via genetic expression. Now that sounds like a mouthful, but here's the basics. Different cells have different shapes and therefore different functions. For example, if we take a neuron with a cell body and all of these tree-like branches coming off of it called dendrites to receive signals, and then a long axon to send the signals to the next neuron. Neurons have this specific shape to communicate or conduct electrical signals. However, if we look at a red blood cell, it has a completely different shape. It kind of looks like a jelly-filled donut or a big saucer with a dent in the middle. This job for the red blood cell is primarily to carry oxygen. So you have two different shapes of two different cells to do two completely different jobs. Conduct electrical signals, carry oxygen, completely different shapes. The genes that are being expressed or turned on in a cell will determine its shape. So the red blood cell got its shape because of the genes that were expressed in that cell. The neuron has its shape because of the genes that were expressed. Obviously, the red blood cell genes were not turned on in a neuron, nor were the neuron genes expressed or turned on in the red blood cell. Genetic expression determines the cell function because the genes code for proteins, and the proteins will affect the structure of the function of the cell. So what are proteins made out of? Amino acids. Obviously, I'm not going to write out all the different amino acids and what the specific amino acids are, but there's 20 different amino acids that make up every single protein for all of life. Bacteria, humans, monkeys, banana trees, your rose bush, everything. All use the same 20 amino acids. The specific amino acids that make up a protein and the sequence of the specific amino acids determine the shape or the structure. So even though all of life uses the same 20 amino acids, they could use a different number of amino acids in terms of how many of the 20 that they use. And they also have a different sequence. Maybe it's the exact same amino acids, but maybe they use a different sequence of the amino acids. It's kind of like, think of the alphabet. All the words in the English language have used the exact same letters. It's just the number of letters and which letters are selected make the difference between all the different words. And then the shape and the structure allows the protein to do its job or the function of the protein. Remember that we have our codon chart from the mRNA codons that will help us to determine exactly which amino acid is going to be put in each spot in a protein. All right, so now let's talk about mutations. We really have to understand all of that stuff about the structure of the amino acids and the sequence of the amino acids determining the shape of the proteins to really understand why mutations are such a big deal. A mutation is any change in a DNA sequence. If it was supposed to go AATT and it got changed to AAGT, well, right there, that's a mutation. Mutations are caused by substances called mutagens. So anything that causes a mutation is a mutagen. There's two main types that we're going to talk about of mutations. The first one that we're going to talk about is point mutations, and the second one is frame shift mutations. But ultimately, point mutation is where a base changes, similar to what we just said there. A frame shift mutation is anytime we add a base accidentally gets added, or anytime we have a base deletion or a base gets deleted accidentally. So let's, for example, let's say here's our normal DNA sequence. Here would be our normal mRNA transcript and the sequence of amino acids. So for this one, let's have a base substitution. Right here, let's put a T in instead of a C. That would change our mRNA transcript, and in this case, notice the first 3 amino acids are the same, but this 4th amino acid here is different than that 4th amino acid there. So we have a different amino acid in that place, which could potentially change the shape and therefore the structure of that protein. Another quick example is if we took that A out. Again, the first 2 amino acids are the same here as they were in the original, but everything after it is changed because we took that one base, this base right here, out if it accidentally got deleted. Again, that would really change the shape of the protein. So the effects of mutations. If the DNA sequence is changed, will the mRNA be altered? Hopefully we say yes. If the mRNA is altered, will the amino acid sequence be different? Maybe yes, maybe no, and we'll see why in a moment. So let's take this example here. Here's our original mRNA transcript. And let's say that there's a mutation right here. We change this U to a C. What would the differences be? Okay, will this change in the code actually affect the functionality of the protein? In order to be able to see if there's actually going to be an effect, let's make sure that we bring up our codon chart and use our codon chart. Well, remember that we always need to start with the start codon, which is AUG. So AUG is down here, and it codes for the amino acid MET. We don't need to know that it actually stands for methionine. AUG. Okay, so now we'll go UUU. It's right there, codes for phenylalanine. GGC. So GGC stands for glycine. And then last but not least, UAG. UAG. Well, okay, that stops. So we're done after these 3 amino acids. So let's see what we get when we changed. AUG is still MET. UUC, wow, right there. We still get phenylalanine. And even if we change again to GGC to GGG, wow, it's still glycine. So here we had an example where we had 2 mutations, but the sequence of the amino acids are the exact same in each case. So the answer to the question, do all mutations cause a change in the amino acid sequence or cause a change in the protein? The answer to that question is no, not always. Can it? Absolutely. Does it always? As we've shown, the answer is no. So now there's different types of point mutations. Remember, before we get into this, though, we said that there's 2 types of mutations. We have frame shift mutations, and we have point mutations. So what we're talking about is the 3 different types of point mutations first. So the first type of point mutation is a silent point mutation. And that's the example that we saw from the last slide. Silent point mutations will not cause a change in the amino acid sequence. Doesn't matter, it's the exact same situation that we just saw. A missense point mutation, missense, will cause a change in the amino acid sequence. And then a nonsense point mutation, it causes a change, but instead of an amino acid change, it causes a stop code. So in other words, instead of having a specific amino acid in there to make that continue with the protein synthesis stops completely. Also, please remember that these are still all point mutations. So let's take a couple of examples. Here's our original mRNA strand and our resulting protein. And let's have a point mutation here. We're going to change that A to a G. So the mutated mRNA strand, again, now we have MET, GLU, ALA, ARG. So we had a point mutation, but the amino acid sequence is the exact same. So this would be a silent point mutation. Here's another example. We'll change that A to a U. Here's the original, here's our mutated strand. What our protein should be here, and after the point mutation, we'll notice that this is a different amino acid than this. So we had a point mutation that caused a different amino acid to be put in. So this would be a missense point mutation. So here's this last one. And so here's our original mRNA strand, and we're going to change that base to a U. Our normal amino acid sequence, just like the others, but our first amino acid, just the same, but when we get to the point mutation here, now that codes for stop, meaning our protein synthesis completely stops, translation's done, none of those other amino acids are ever going to get made. We stop protein synthesis. So this would be a non-sense point mutation. So those are all the point mutations. Now let's talk about the other type of mutation. It's a frame shift mutation. A frame shift mutation is, like we said before, is when you either add or delete a base or a nucleotide from DNA or RNA. This will cause the mRNA codons to change at the spot of the mutation and every codon after that. So for example, we know the ribosome is looking for AUG. And then it's going to look at those 3 base pair sequences after the start codon. So there's a codon, there's a codon, there's a codon, there's a codon, and UGA happens to be a stop code, so we know it's stopped there. However, let's say that we take out this base right here. This base is deleted. Well, now, instead of having our codon here, the codon would be there, right? 1, 2, 3. And then 1, 2, 3, that's the next codon. 1, 2, 3, that's the next codon. 1, 2, 3, which is GAG is not a stop codon, and it would probably continue to go forever and ever until it got that stop codon somewhere randomly down the line just by chance. So if you notice that reading frame, or where the codons are that we originally saw where they're supposed to be in white, because we deleted a base, that shifted the reading frame or shifted the codons in a particular direction. So now the codons that's supposed to be there are not there at all. They're completely different codons, which means completely different amino acids and different proteins. Frame shift mutations, by far and away, are the mutations that can cause the most damage to a protein in terms of what it's supposed to do and what it actually does. So here's another example of the frame shift mutation. Here we take out the G, which slides everything else down. So instead of having methionine and then GLU, now we have methionine LYS, HIS, and it's completely different. Who knows what's going to happen after there? I didn't want to put in a full actual protein because that would make our video too long. The sequence of amino acids determine the shape and the job of the protein. A change in the sequence of amino acids changes the shape and therefore the job of the protein. Mutations can change the sequence of amino acids. There's 2 types of mutations: point mutations, where we substitute a base, and frame shift mutations, where we add or delete a base. And that is all she wrote.
It's like this and like that and like this and it's like that and like this and like that and it's like this. So just chill till the next episode.